Transgenic models are organisms used by researchers to study the nervous system by selectively altering gene expression. Neuroscience relies on animal models to study complex processes that we would be unable to investigate in humans. The fruit fly, zebrafish, and rodents are commonly used model organisms in neuroscience research. Of these, the mouse is widely used due to the genetic and physiological similarities to humans. There is also a wealth of information available about the genome of model organisms, which allow scientists to alter the genome and thus create transgenic organisms. The term transgenic generally refers to genetically altering the genome through the introduction of DNA from the same or different species. For example, a certain species of jellyfish emits a natural glow or bioluminescence. Scientists have extracted the light-emitting property from the fish and then inserted it into the genome of bacteria and other model organisms, allowing tissues to fluoresce green when excited with ultraviolet light. The protein, named green fluorescent protein, or GFP, is now a common tool used for easy visualization of specific targets. The addition of green fluorescent protein into the genome is an example of one type of transgenic model used in research, knock-in models. Knock-in mice contain a gene or gene mutation that is inserted into the genome. Commonly used transgenic models are called knockout models. Knockout refers to the deletion, inactivation, or silencing of a desired gene. This can provide a wealth of information regarding the function of a gene. For example, if you knock out a gene important for promoting water intake, mice may not drink a normal amount of water and become dehydrated. The transgenic animal used will depend on what question researchers are trying to answer and what organisms would serve as an appropriate model for investigation. While there are many different transgenic species available for research, here we will be focusing on transgenic mice, how they're created and utilized in behavioral neuroscience. So, how are transgenic mice created? The creation of these mice is a multi-step process. First, the target gene and its location in the genome is identified. One advantage of widely used model organisms is that their complete genetic code is already known, allowing for easy targeting and manipulation of the genome. Once the target gene is identified, it is isolated from the host organism. This is a multi-step process that involves isolating the DNA from tissues and using polymerase chain reaction, or PCR, to create many copies of the target gene. Following isolation, the genetic sequence is altered in the lab using complex genetic engineering techniques. After this genetic engineering, the newly synthesized gene needs to be inserted into the genome of a host animal. This is done by inserting it into a fertilized embryo of the model organism. The altered gene will then incorporate into the embryonic genome. Commonly, the altered genetic sequence is created to have portions of DNA that are identical to the embryonic DNA. This allows the cells to recognize these sequences and splice the altered DNA strand into the genome, thereby replacing its original genetic sequences. To create a knockout model, scientists can insert a stop codon into the genome, preventing transcription of the target gene and therefore resulting in a silencing of the target gene. The sequence can also be excised from the genome so that after homologous recombination, the target gene is removed. Following gene insertion, the embryos are transferred to a surrogate female mouse that will eventually give birth to the first generation of transgenic mice. Colonies are set up to produce offspring of a specific genotype needed for experiments. 2 copies of the altered genetic sequence are required to produce a functional change in the animal, one on each chromosome. Wild-type animals are those that do not contain the altered genetic sequence. Heterozygotes are offspring that carry only 1 copy of the manipulated gene, and homozygotes express 2 copies of the altered genetic sequence. Therefore, a mouse that contains 2 copies of a silenced gene will not express that gene in any quantity, whereas a heterozygous mouse would still express the target gene. Commonly, wild-type mice are used as a control group, as they have no copies of the altered gene and therefore will display normal physiology and behavior. The experimental group is the group of mice that receives the treatment, or in this case, the group that contains the altered genome. Therefore, homozygous mice are commonly used as experimental subjects to study how gene insertion or deletion impacts behavior. Since heterozygotes only contain one copy of the manipulated gene, they can be utilized in experiments that study partial gene mutations. Once the in vitro genetic engineering occurs, the resulting offspring will be maintained in a breeding colony. Genetic engineering is a time-consuming and costly process, so scientists need to be able to produce transgenic mice without having to repeat the engineering in vitro. The mice are bred to produce offspring that carried the altered genetic sequence, and subsequent mice can then be used in research when they reach the desired age. Heterozygotes are also important for maintenance of a transgenic colony and frequently used as breeders for future generations of mice. Let's say, for example, that researchers want to study how a particular gene is involved in regulating stress and anxiety. To do so, they decide to use a transgenic mouse that is a knockout for the corticotropin-releasing hormone that is important in coordinating the stress response. To study the effects of this gene, they test the mice using the elevated plus maze. The maze has two arms that are completely open, with no barriers on either side, and two arms that have dark walls lining the arms. As mice are prey animals, they naturally favor dark and enclosed spaces, rather than open areas. Researchers can compare the behavior of the wild-type mice to that of knockout mice to understand how the corticotropin-releasing hormone impacts behavior in anxiety-inducing situations. A heterozygous mouse may be used to study how a reduction in gene expression changes behavior, and this may be more representative of genetic differences that occur in humans. Therefore, the use of transgenic mice is a valuable tool for scientists to isolate and study the effects of specific genes on physiology and behavior.